Good morning to all. On behalf of the Department of Biochemistry, Indian Academy Degree College Autonomous, Bengaluru, I, Dr. Suma TK, extend a hearty welcome to one and all present here to today's special webinar on the occasion of World Cancer Day. This cancer awareness webinar titled Anti-Cancer Drugs from Mother Nature, Ancient Solution Biochem Club of the Department of Biochemistry, IADC-A, in association with Institutions Innovation Council, MHRD, Government of India. I welcome our respected chairman, Dr. T. Somashekar, and our principal, Dr. Srinidhi K. Parthasarathi, to this webinar. It is to extend a warm welcome, University of Mysore, Manasa Gangotri Campus, Mysore, who is going to enlighten us all on the topic of anti-cancer drugs from nature. Welcome, sir.
Thank you very much.
I also welcome our head of the department, Dr. Anandavardhan H. I also extend my hearty this huge victory of and students with immense pleasure. The global cancer community every year with the purpose of So February 4 every year is organized as the World Cancer Day with the purpose of creating awareness to inspire change and reduce the impact of cancer. It's an initiative aimed to achieve greater attention towards cancer, its prevention, detection, and treatment, and also to reduce the social stigma associated with the disease. A small step towards this objective is this webinar organized with the simple intention of improving awareness among people, sensitizing the public also about this important issue, inspiring action, and also encouraging all young future and current scientists. Now, before starting the webinar, I request everyone to maintain decorum throughout the session and remain muted. Any questions can be put down in the chat box, and it will be read out by the host at the end of the speech, awaited part of today's program, which is a talk by Dr. C.D. Mohan, NASI scientist of University of Mysore, who is going to nature ancient solutions for modern problems. I now request Dr. C. Naveen Kumar, Assistant Professor, Department of Biochemistry, IADC-A, to
Thank you, madam. Good morning, and I welcome everyone for Cancer Awareness Webinar. I am Dr. C. Naveen Kumar from Department of Biochemistry, IADC-A. It's a great pleasure for me to introduce our speaker today, who is going to talk to us about anti-cancer drugs from Mother Nature, ancient solutions for modern problems. Our speaker, Dr. C.D. Mohan, he is an assistant professor in the Department of Studies in Molecular Biology, scientist at Adi Chunchandgiri Institute for Molecular Medicine, BG Nagara Nagamangala Taluk, Mandya. He did his PhD in biochemistry under the supervision of Professor K.S. Rangappa from University of Mysore. His research oriented towards designing of synthetic small molecule inhibitors against pro-inflammatory signaling pathways in cancer. And he discovered several inhibitors towards JAK-STAT, BAD, TNF-alpha, and NF-kappa B in cancers. Dr. Mohan has published 71 research papers in international peer-reviewed journals, including Proceedings of the National Academy of Sciences, USA, Journal of Biological Chemistry, iSciences, Scientific Reports, etc. And he has 5 patents in the area of cell biology. He has been awarded Jubilee Award by the National Academy of Sciences, India in 2019, Professor Umakant Sinha Memorial Award by the Indian Science Congress Association, Kolkata in 2020, PHSS Young Scientist Award by the Professor H.S. Srivatsava Foundation, Lucknow in 2022. Recently, he has been conferred with Biochemie Article of the Year Award by French Society for Biochemistry and Molecular Biology and Elsevier with a cash prize of €1,000. He has been invited to deliver a talk at the annual Congress of French Society for Biochemistry and Molecular Biology, which will be held at University of Paris, France on 4th or 5th July 2022. So dear participants, please join me in welcoming our eminent speaker, Dr. C.D. Mohan. Please welcome, sir. Now this session is open for you.
Thank you very much, Naveen, for introducing me to the wide range of audience that you have. Like I can see that there are 203 participants. It's a quite huge number, in fact, for the webinars. And I thank Dr. Suma also for welcoming me for this webinar. So initially, to tell about myself and Naveen's association, we both are very good friends from very long time, and he worked with me in my department previously. Later he shifted to the Indian Academic College, and of course when he decided, when the management of the college decided to celebrate the Cancer Awareness Day, he spoke to me that I might be the good speaker to have on the board during this on this day, and I'm happily accepted because of my close association with him. And let me speak to you some critical things about cancer. How exactly today is Cancer Awareness Day, World Cancer Awareness Day, and let me explain you how exactly cancer is impacting our population, or mankind, in fact, throughout the globe. So next 30-40 minutes, I'll just give you some critical information. I think I can share my slides, which have been prepared. So can you see my slides? Can someone confirm that? Naveen, can you see my screen?
Yes, sir. Yes, sir. I can able to see.
Okay. Thank you.
Make it full screen, sir. Make it full.
So, hello guys. Today I'm talking to you about anti-cancer drugs from Mother Nature, ancient solutions for modern problems. I'm calling it the cancer as a modern problem. I'm referring it to cancer. And ancient solutions means I'm going to talk to you for first few minutes about what is the basis that we are getting from the traditional knowledge using the traditional medicine. Like it may be Ayurveda, Unani, or Chinese traditional medicine. We have different kind of medicinal system throughout the globe. Are these chemicals, are the medicines that are obtained from these traditional medicine system, how they are going to impact the cancer in the human system? So that's why this is the relevance to the title. In fact, let me tell you, I should, without giving the statistics of the cancer, you may not be able to follow what is the impact of cancer on the human society. If you look at this slide, this is the most recent statistics available on the WHO website, World Health Organization website. As per the WHO report, the top five the cancers affecting the human health throughout the globe are lung, colorectal cancer, liver cancer, stomach cancer, and breast cancer. So these are considered as the top five cancers in terms of death rate. You can see that there is a pink box which indicates the number of deaths took place in just 1 year, 2020. So it is 1.8 million people dead due to lung cancer, 0.9 million due to colorectal cancer, 0.83 million due to liver cancer, stomach cancer. It contributes to 0.769 million deaths, and breast cancer is approximately 0.685 deaths. In the same way, the blue box indicates the new cases that were reported in 2020 alone. So if you look at the number, it is everything in millions. So cancer is one of the leading causes of death throughout the globe. It is after the heart related problems, cardiac arrest, cancer is the highest disease that is affecting the global death rate. Therefore, it is very essential to have a precaution. I would say the precaution to prevent this disease. So first, let me tell you what is cancer. When some cells begins to proliferate, begins to divide enormously in your system, in any of the organ, we call it as cancer. And this is due to the enormous proliferation, uncontrolled proliferation of the cell is taking place due to various reasons, which I'm going to discuss in the future slides. But very alarmingly, what I would like to bring it to your this panel is the number what I have shown here. The number is going to be doubled by 2030. That means by another 10 years, if I have written lung cancer 1.8 million deaths, it is going to reach around 3.6 million deaths in 2030. So why we are moving so fast towards death through the cancer? The question that arises is why the cancer in olden days the cancer related deaths were very less, but why, what is causing the problem to be so enormous, and why the death rate is going to be increased so much in next 10 years? The problem is here. Let me give you some examples and explain you why we are prone to cancer easily. So this is whatever the slide I'm showing you here now is in the global context. In case of the Indian context also, so very recent information is not available in the related to Indian population, but the data presented here is taken from 2018 statistics. So as per the statistics, breast cancer stands first, and oral cancer stands second, cervical cancer stands third, lung cancer stands fourth, and stomach cancer stands fifth. The all other contributes to the remaining type of cancers. And you should also know that in the global context, 24% of the cancers are due to the lung cancer, and other 76 is contributed by different types of cancer. So there are totally more than 200 types of cancers identified so far. So breast cancer is considered as one type, oral cancer, lip cancer is considered as one type, Like this, 200 different organs can be affected, and that may lead to cancer. So it is really you name any organ, that organ can be affected with cancer. Let me give you, this chart which explains you what are the reasons for the cancer. The first thing is exposure to carcinogens. See, we are under the impression that we are not getting exposed to carcinogens. First, let me tell you what is a carcinogen. Carcinogen is any chemical that turns the normal cell into cancerous cell, is called as a carcinogen. So as I explained to you in my previous slides, we are getting exposed to more amount of carcinogens day by day. If you don't believe my words, I'll give you some instances. See, in case of in fact, our chairman, like in our department, we have a practice that in the fourth semester, MSc students should take part in a research project program. That is a minor project that they do it in our department in the fourth semester as a part of their academics. In our chairman's laboratory, Professor Gopal Marat's laboratory, they wanted to identify whether the food that we are eating are adulterated or not. So in this case, they took so many the ingredients from the local market as well as from some branded shops and tested for the adulteration. The first thing what they did is they took green peas from the local market in the Mysore, and they took the same green peas from Big Bazaar, Loyal World, and other branded shops like this. When they tested for the adulteration, we got it. They got the results. They reported this also. These green peas were coated with a chemical called as malachite green. As you are aware that malachite green is a potent carcinogen. Just to make them appear green, the freely available the green peas were coated with or mixed with malachite green, which is causing a hazardous condition in the society. If you go to eat snacks in a roadside vendor, he would prefer to take the green peas available in the local market, which is much cheaper than the branded shops. Therefore, this acts as a media of transmitting the large amount of mutagens to the society. So carcinogens are introduced into the society. So just you are unaware that what you are eating through green peas are mixed with some chemicals like this. In the same way, since the students, here there is a lot of students in this one. So during your experimentation, I'm very sure that nobody uses gloves. So that means the chemicals that you are using during your experiments, you never know the chemical nature of those chemicals. They may be a potent carcinogen. There may be a potent carcinogen or some mutation related mutation causing chemicals. Therefore, students are never unaware that they are touching the chemicals with their bare hands. So if, for example, ethidium bromide, using of the ethidium bromide is very, it is a potent carcinogenic agent. Students use it with their bare hands. In the same way, see, most often we all have the habit of having coffee or tea in plastic cups. So these plastic cups are made up of a chemical called as melamine. So melamine is a kidney carcinogen. Nowadays, you can see that there is an increased number of kidney related issues. The patients, you can see if you go to multi facility hospitals, you can see that the large population are suffering from the kidney disorders or kidney failure. So that is due to melamine, which we don't know that we are drinking coffee or tea. No, in fact, we are drinking coffee and tea along with a chemical called as melamine, which has a serious threat to the life, and as well as it causes the disease. So the first point says it is exposure to carcinogens. I just took 2-3 examples to tell you, since it is a Cancer Awareness Day, I feel that this is the right platform to educate the people so that they know what to eat and what not to eat. So exposure to carcinogens can be prevented if you start avoiding such things. There are hundreds of instances where we eat this kind of chemicals every day. Other than that, radiations. The next comes is the radiation. Do we really get exposed to radiations? Yes, we do. It could be through natural phenomena, or the UV light that that falls on us from the sun, or could be the artificial phenomena. I mean to say, so the last 2 years we have been witnessing the COVID has taken toll in throughout the globe. In this condition, the people are advised to undergo CT scan regularly. Regularly in the sense, probably that has become a confirmatory test. You should know that X-ray itself is very hazardous. Getting X-ray, if you are getting chest X-ray, you have chances that that some organs present in that area may become cancerous. So it is highly recommended not to have X-rays unless there is a serious issue to be diagnosed. But CT scan is equal to getting one CT scan is equal to getting 200 X-rays. So CT scan is so much hazardous. Therefore, in the COVID situation, most of the COVID affected patients underwent CT scan. Therefore, it is highly expected in the near future that increased amount of CT scan may result in the increased number of cancer patients in next 2-3 years. See, please don't be under the impression that those who have undertook CT scan will get cancer. It is not that way. CT scan is a potential hazard. Just I'm telling you, CT scan is a potential hazard. And one more thing is, 15,000 Americans die due to cancers arised by CT scan. This is a data available in the scientific sites. You can verify this. Therefore, since in the Indian system or in the underdeveloped countries or in developing countries, we don't have the records maintenance in such a way that we may not know where exactly what is the impact of these radiations on the human health. But CT scans are hazardous. Please keep this in mind. Then next one is bacterial and viral infections. The slide says bacterial and viral infections. So in case of the bacteria, I can take an example of Helicobacter pylori. Helicobacter pylori is a immigrant of the human gut. So it it tries to resides when it infects a person. It tries to resides in the stomach. So in the stomach, it start corroding the inner wall of the stomach, which eventually leads to cancer. Thanks to our Indian food system, we are not susceptible for Helicobacter pylori infection due to the excess amount of spices that we use. The spices that we use have the active principles which controls the infection related to Helicobacter pylori. Therefore, but this type of infection is very common among the Westerns. See, when I listed the top 5 cancers, stomach was also there on among them. That stomach cancer is majorly contributed by Helicobacter pylori infection, which is common in Westerns and not uncommon in the Indian population. In the same way, the viral infection, it's also written the viral infection. All the virus cannot cause cancer. Only few cancers can be caused by the virus. For example, HBV. I'm very sure that you are all familiar with the hepatitis B virus. Hepatitis B virus is a DNA virus. These DNA viruses are dangerous. They can incorporate their genomic DNA into the host cell, and it may result in the cancer. So it is advised, as soon as the hepatitis B virus is in, if the person get infected with hepatitis B virus, he should be treated immediately and try to treat that person with a particular treatment regimen so that the person recovers quickly, and most of the viral content will not stay back in the system. So the bacterial and viral infections are again can be the reason for the cancer progression. Then the next one is smoking. As you all know, smoking is considered as one of the potent causative agent for cancer. Here, one thing is, if you have a smoker friend, I suggest you to keep him away, because once the smoking is the smoke is left from the smoker, he is releasing 66 mutagens into the system. So once he leaves the smoke, it is releasing 66 mutagens into the surrounding. So if you inhale, as a friend, if you inhale, though you are a non-smoker, if you inhale those 66 mutagens as carcinogens, there are a very potential chances that these mutagens can cause cancer. So it is better to not. It is better to keep the friend who is a smoker away than having a smoke. Then the next one is poor diet. Of course, the poor diet in the sense we eat nowadays lot of processed foods. These processed foods are enriched with the chemicals which are accident in nature, which generates a lot of free radicals in your body. So you may experience, if you eat burger in some big restaurants, you will experience constipation the next day. It could be due to the addition of the processed chemicals. So therefore, poor diet also contributes to the generation of this cancer. And apart from this, as I explained you already, aflatoxins, it could be a hepatitis B infection, hepatitis C infection, aflatoxins. All these are the contributing factors for the development of the cancers. So people should know cancer. The agenda of celebrating Cancer Awareness Day is the large amount of people should be educated what to be done and what not to be done. I strongly suggest you. So if you are using some chemical or some, it could be your, cosmetic agents, or if you are eating some food, please be aware of what you are eating and what you are drinking. See, you never know the colored food. You eat gobi manchuri, so you never know what is the color agent that they have used there, and what is the impact of that coloring agent on your, health. So please be careful about this one. These are the reasons why the cancer is growing in an alarming way. I said the statistics. I told you about the statistics, and it is going to be doubled in in next 10 years. This says that there is a immediate need to cut down or modify our lifestyle in such a way that we go it in we carry our lifestyle in a better way. So speaking some again about the cancer, in case of the normal cells, in the upper panel you can see that the cells undergo division. One cell becomes 2, 2 becomes 4, 4 becomes 8. In the exponential form, the cells become the cells grow. So in case of the normal human being, we all eat some mutagens or we get exposed to some carcinogens. When we get carcinogen, the cell get damaged. The damaged cell undergo apoptosis. Immediately it is recognized by the our system and it undergo apoptosis. Apoptosis means the cell will be killed. In case of the how cancer originates is, if there are mass of cells, if they experience some chemical mutagen, they undergo mutation repeatedly. When the cells undergo mutation repeatedly, they escapes the surveillance mechanism, and which turns them into a cancer cell, which grows enormously. So basically, cancer is originated due to mutations in two set of genes called as either in the oncogenes or in the tumor suppressor genes, which I'm going to talk to you about in the later stages. So cancer are broadly classified into two types: benign cancer and the malignant cancer. Benign cancer is there are some cells which undergo mutation, but those cells are restricted to some area. They grow there, but they cannot spread from one place to another place, and they can be easily treated by simple operation. Those cells can be removed, and it is called as benign type of cancer. The other type of cancer is malignant type of cancer. It is the metastatic in nature. So in this picture, you can see that the primary cancer has been taken place in the colorectal region or in the intestine. Once the cancer cells are formed in the intestine, they begin to proliferate, they make a tumor mass. Once after the mass is formed, they enter either through lymphatic system or through blood vessels. They enter and they begin to search for a favorable condition. So if they find a favorable condition in the lung, they go and sits there and begins to proliferate. We call it as the secondary tumor. Or if they find it in the brain, if they find favorable condition in the brain, they goes and sit there and begins to proliferate there. We call it as the brain metastasis. So basically, this is what happens as the cancer advances. During the primary conditions, they begin to proliferate in an initial one organ. If the cancer is diagnosed at this stage, cancer can be cured. If the cancer cells metastasis takes place to different organs, then probably the cancer cannot be cured. So in the first two stages of cancer, cancer is restricted to only one organ. In the third and fourth stage, they undergo metastasis. That means that you can see in this picture that the cancer cells have entered the blood circulation and passing on to the next organ. So in that condition, it is unlikely that the cancer can be treated. So coming to more into research aspects, cancer cell has all these properties. The heading says cancer cells are immortal. First, I should tell you what is cancer immortal condition. What is the causing it to be immortal? Immortal means the cells have no death. These cancer cells never die. I'll tell you a simple example to make you understand how exactly these cancer cells can survive for how many number of days or years. Generally, in all our laboratories, those who are working in the cancer, we grow cancer cells in the laboratory. So once we procure from the IADC-A, we grow for enormous number of cycles in our laboratory. For the first time, cancer cells were grown in 1951. So till 1951, people were trying to grow cancer cells in the laboratory conditions, but they were unsuccessful. In 1951, the cancer cells were extracted from a female and they started growing. Let me tell you about the female also. African as well as American female. She's a Negro woman. Her name is Henrietta Lacks. She comes with a complaint to John Hopkins Hospital at USA, stating that she has a severe stomach pain. When the doctor diagnosed her, he found out that the lady is suffering from cervical cancer. Without her notice, he collected some cells without her notice as well as consent, which is unethical in today's phenomena. So without her notice, he collected some cells from her tumor and put it in the media that he had in his laboratory. Fortunately, this is the first success that they got. The cancer cells begins to proliferate. So as I said, cancer cells were never cultured before. It is in 1951 for the first time cells were cultured in the laboratory condition, and those cells begins to proliferate in the culture conditions. Then unfortunately in 1951 itself, the Henrietta Lacks died, but those cells were taken from Henrietta Lacks. They were named as HeLa cells. They were named as HeLa cells. HeLa. He means Henrietta, La means Lacks. So it was, as I said, it was isolated in 1951. Today is 2022. Still, the HeLa cells have been used as a gold standard cells throughout the globe. In all the laboratories who are working with the cancers are using HeLa cells throughout the world. So what I meant is, HeLa died in 1951. Even after 75 years after her death, still we are using HeLa cells in our laboratory conditions. So this is what the meaning of the cell immortality. Those cells never die. They have no death at all. So cancer cells have enormous proliferative potential. So you can see that they have they prepare their own cytokines, they prepare their own signaling molecules, and they have sustainable proliferative signals. They have enormous replicative potential. Replicative potential means they can divide any number of times. So HeLa can be preserved probably for another 500 years, I would say. So no contact inhibition. Generally, the surrounding tissue controls the proliferation of the cell. In case of the cancer cells, they do not respond to the negative signals, and they do not respond to the contact inhibition. And they have the property of invasion and metastasis. Kind of nature and apoptotic resistance. Every cell should die after a certain amount of time. So this, in this case of this one, the cancer cells have apoptotic resistance. They never die. It is something like this. So it is highly essential to have some strategy to kill these cancer cells to treat the patient. So we consider this as a cancer care or cancer therapeutics. So cancer can be treated through 5 different ways. As per the current understanding, cancers can be treated through 5 different ways. One is through surgery, the other one is through radiotherapy, the other one is through cytotoxic chemotherapy, the next one is molecular target therapy, immunotherapy. These are the 5 pillars of cancer therapy. So among these 5 pillars, our laboratory works are also with synthesize some chemicals that have the anti-cancer effect towards the cancer. That our research area focus on these 2 things: drugs either from the natural sources or drugs from the synthetic origin. See whether, like as I said in the initial slide, our Ayurveda, Unani, or some other type of traditional medicine system has provided us a lot of information about the drugs that are available in the nature. I would say Taxol, Vinca-alkaloid, camptothecin, podophyllitaxin are all the drugs that are used today for the treatment of cancer are derived from the natural sources or from some plants. In the same way, sorafenib, gefitinib, methotrexate are the drugs that are synthesized in the laboratory. They are not from the plant origin, they are from the laboratory. So both synthetic origin as well as the plant sources have been contributing for the source of as the source of drug to infect or which is to cure cancer. So I'll just tell you a very interesting story about the production of the Taxol. See, initially this dates back to 1962. In 1962, National Cancer Institute of USA funded a project to some scientists of their country. Their intention is to find some chemicals or some drugs from the plants. So they went on profiling several plants, searching for the new anti-cancer drug. They visited a tree called as yew tree. The name of the tree is yew tree. The scientific name is Taxus brevifolia. This yew tree, when they took the bark of the yew tree, the bark was taken to the laboratory and grinded and started testing on the cancer cells. They found a very interesting result. Yew tree extract of the yew tree killed the cancer cells completely, in fact, completely. And they were astonished by the results and tried to know what exactly present in that yew tree, bark of the yew tree, where it is, since it has a good anti-cancer effect. When they crystallized the molecule and found out the structure, they got to know that there is a presence of a compound called as Taxol in the bark of the yew tree. This is the structure of the Taxol. So when it is tested on the cancer patients, it worked amazingly well, and that was introduced into the market in 1975. So Taxol, which is a this took place in 1962, but very important point that they noticed here is the Taxol content is so low in the yew tree. The Taxol content is approximately 0.01% to 0.03% in the bark of the yew tree. Therefore, it was very difficult. Let me tell you, the last statement says the amount of drug, that is the amount of Taxol required to treat a single cancer patient is approximately 2.5 grams. To get that much amount of Taxol, we need to cut 6 yew trees of 100 years old. So you can assume the availability of Taxol in a very small amount in each tree. To treat single patient, we have to cut 6 yew trees. So due to this one, it leads to lot of deforestation of yew tree. So this went on till 1993. I would say, Taxol is in the present day concept also. Taxol is the leading seller, and it is the world's first billion-dollar anti-cancer drug. So we found out, I mean, the scientists found out an alternative method to produce Taxol in 1993. But between the 1962 to 1993, lots of yew trees were cut down. See, these are the two people who showed an alternative method for the production of the Taxol. See, what they did is a very simple experiment. They took the bark of the yew tree. That bark was kept in there, since they are microbiologists, they are kept the bark in the Petri plates, that is nutrient agar Petri plates. And around that bark, some fungus started growing. So what they understood is the bark has some fungus in it. So when they grow that fungus, they extracted all the chemicals produced by the fungus. Later, they found out that the fungus is producing the Taxol. So it is like maybe through horizontal gene transfer or some other way, the plant is also producing Taxol. The fungus is also producing Taxol. It is unsure that from where the genes have been contributed, either from the fungus to tree or tree to fungus. So what they have found out is a fungus associated with the yew tree can produce the Taxol. Then the bottom line is fungus can be grown in your laboratory. I'm telling you, your laboratory in just 15 days. So for the production of Taxol, we require 100 years under a tree here, but we can just produce the same amount of Taxol in 15 days. So this is how these people, Donald and Sterl, discovered a way to produce the Taxol in very easily. In the same way, so once after the discovery of the Taxol, in the same way, multiple researchers started working on these medicinal plants. You may be aware of the Vinca rosea, which is present in the gardens of almost all the houses. So Vinca rosea also has the anti-cancer property, and later, in the same way, they isolated the plant, they crushed it, they found out that there are the alkaloids present in the Vinca rosea, and vinblastine is one of the alkaloid that is present in the Vinca rosea, which has been used as an anti-cancer drug in today's market. Like this, several drugs were discovered. Maybe camptothecin, podophyllotacin, all these are available from the plants. So what we understood from these things is Mother Nature is serving as the source of anti-cancer drugs, so that we need to explore more amount of drugs from Mother Nature. Then only we can we will have a better drugs in the near future. So after explaining you some basic things about the how to target the cancer, or what are the properties of the cancer cells, and how do you get cancer, now I'll orient myself towards the research that we are doing in today's in our laboratory. So basically I work with targeting STAT3 signaling in cancer. So STAT3 signaling, probably the higher semester students that are there in the presentation may know what is STAT signaling. So I'll just explain you about STAT3 signaling. Later I'll move on to the how we design drugs, and you will be able to follow it. So this is what the STAT signaling works like. So in the picture you can see that this is the plasma membrane of any cell. It could be plasma membrane of the any cell. There are receptors which are called as GP130 receptors. These receptors, when the cytokine binds to the receptor like this, there is a protein called as JAK. JAK means Janus kinase. Janus kinase protein is a non-receptor tyrosine kinase which is present in the cytoplasm. So Janus kinase could be, there are 4 types of Janus kinases: Janus kinase 1, Janus kinase 2, Janus kinase 3, and tyrosine kinase 2. So Janus kinases are present in the inactive form with the receptor in the cytoplasm. When the cytokine binds to the receptor, Janus kinase get activated. Activation here I'm referring to the phosphorylation. Janus kinase get phosphorylated. In the inactive form, they are in the unphosphorylated form. So when they get phosphorylated, they get activated. So the function of the Janus kinase is, as you know, it is a kinase. Kinase phosphorylates its substrate. Its substrate is STAT protein. So STAT protein is present in the monomeric form in the cytoplasm. When the Janus kinase get activated, Janus kinase phosphorylates the specific tyrosine residue on the STAT protein. Once the STAT protein get phosphorylated, they undergo dimerization. You can see in this picture that the STAT protein undergo dimerization through the phosphorylation. So once the dimer has formed, it enters the nucleus through using impotins. It enters the nucleus. You can see that in the nucleus, STAT is a transcription factor. Transcription factor means it binds to the specific DNA elements and expresses the genes. So STAT is a transcription factor which is responsible for the transcription of several genes. I'll talk to you about the genes later. So parallelly, I should also tell you that there are 7 different types of STAT proteins: STAT1, STAT2, STAT3, STAT4, 5A, 5B, and 6. So these are the different types of STAT proteins that are present in the cell. So I'll show you an interesting table. Here, this table is taken from the Journal of Clinical Investigations. So the table says that some STAT proteins are overactivated in some types of cancers. So it can be breast cancer, head and neck cancer, prostate cancer, melanoma, or different types of cancers, as also from leukemias or blood related cancers. You can see that there is activation of STAT proteins 1,3, 1,3, 3, 3, like this. Some particular type of STAT proteins are activated in the cancer cells, but majority of the cancer shows STAT3 activation. You can see that there is a presence of 3 in the activated variant. So STAT3 protein get activated in case of the cancer. Therefore, if you can inhibit the STAT3 activation through some mechanism, we can counteract the proliferation of the cancer cells. So this is our research idea. So we wanted to know. So in this picture, you can see that there is a presence of the interleukin 6, a particular cytokine. There is activation of the JAK proteins. Then the STAT3 undergo phosphorylation and dimerization. It enters the nucleus. So what we are interested is When it enters the nucleus, it transcribes the genes that are involved in the cancer causation. Therefore, we want to prevent the dimerization of the STAT3. If the dimerization of the STAT3 is hampered, then it cannot enter into the nucleus. The monomeric STAT3 cannot enter into the nucleus. So this is our research idea, to stop the entry of STAT3 into the nucleus. So what we did, I'll tell you what we did in our laboratory. So this is how the project designing was done. Probably the project designing goes above the head to the degree and postgraduate students. I'll skip this slide. Here, when we searched through the literature, we found out that there is a drug molecule called as atiprimode. Atiprimode comes under a category of azaspirines. Azaspirines means, you can see that there are two rings. Two rings have a common carbon atom. You can see that at this point, this kind of ring is called as spirine ring. Spirine ring have a single carbon atom which is involved in two ring formation. So this is unusual, very unusual. So what the preventer inventors, that is Dagger and et al. Did is, they discovered this atiprimode, and they showed that JAK2 is inhibited by atiprimode, and the concentration is given here. In multiple myeloma, they showed the JAK inhibition. It is in the phase 2 clinical trials in the today's market, and this molecule has been patented by Dagger and colleagues. So what we decided is, since atiprimode is a promising drug candidate, what we did is, we wanted to synthesize a very similar molecule which doesn't cover under the patent. So what they did is, they used 6-membered ring and conjugation with 5-membered ring. What we did is, this is our scaffold. So there is a 6-membered ring conjugation with the another 6-membered ring. So the same spirine nucleus, we have conjugation between 6-membered and 6-membered rings. So this generated a new molecule. So the basic idea is, as I explained you, we are trying to target cancer through designing a new drug. So in this project, we have synthesized a new molecule which has two 6-membered rings. Then we wanted to know, we chemically derivatized. So using you can see that all the rings, all the chemical compounds have two azaspirine rings. We chemically derivatized it and tested on the different cancer cells. First cancer cell line that we evaluated is HepG2. HepG2 is a hepatocellular carcinoma cell. Hepatocellular carcinoma means liver cancer cells. So we tested all the compounds against these liver cancer cells. So you can see that there are 10 compounds, and this is the IC50 value. IC50 value is, here we have synthesized 10 compounds in our laboratory. When we put those compounds into the HepG2 cells, how many cells are dead? The amount of drug required to kill 50% of the cells is considered as IC50 value. We found out that the drug named 5F, or I'm calling it as CIMO now onwards, worked very well, and it showed the cytotoxicity at 7.3 micromolar concentration. That means the drug CIMO was killing 50% of the cells at 7.3 micromolar concentration, and this is the very good results, in fact. And we carried out the assays further. We wanted to know, whether CIMO has effect on the normal cells or not. It is like in the cancer treatment regimen, it should be considered that when the if you get a cancer drug, you should make sure that the drug doesn't kill the normal cells. If the compound kills the normal cells also, there is no point in using that drug. So what we did is, we used a cell called as LO2. LO2 is normal hepatocytes. So normal hepatocytes were treated with compound CIMO, and it was not active till 100 micromolar. So it is a very promising result. Our drug specifically targets the CIMO, the cancer cells, not the normal cells. Then we carried out the next assays. Next assay is, you can see that we tested again the SIMO on different types of cell lines. You can see that liver cancer, gastric cancer, breast cancer, prostate cancer, and head and neck cancer. And you can see that there is a IC50 value between 3 to 7 micromolar. So what we understood is this SIMO molecule is working well with against all the different types of cancer cells tested. And one important thing that we noticed is it inhibited a particular cell line called as BT549. BT549 is a breast cancer cell. To be more specific, in the breast cancer, there is a subtype called as triple negative breast cancer. See, if there are 100 females suffering from breast cancer, among them, 15 females are suffering from TNBC. The percentage of TNBC is triple negative breast cancer is 15%. Generally, it is 15%. That means TNBC means those females, they do not express 3 proteins. One is estrogen receptor, progesterone receptor, and HER2 receptors. So these 3 are the targets of our drugs, presented drugs. So if what those cells does is, the cancer cells does is, they stop expressing these 3 receptors. If these receptors are not expressed, those cancer cells cannot be killed. So 15% of the women suffers from TNBC, where we do not have the medication till now for the treatment of the TNBC. So very fortunately, our drug showed good activity against TNBC cell lines also. But our primary target is, we wanted to know whether it inhibits STAT3 phosphorylation or not. Because as you know, whenever in case of the cancer cells, STAT3 is overactivated. So you can see that compound 5F, or in the other way, it is called as SIMO, showed good inhibition towards phospho-STAT3. So which means there is a clear area, which means that there is no STAT3 phosphorylation. So indicating that the STAT3 undergo the phosphorylation of the STAT3 has been hampered by our SIMO molecule. And we tested against FG2, AGS, PLCPR5. We found the same results with the concentration gradient. The cancer cells were dead because of the concentration gradient of this our drug. So we performed flow cytometric analysis. You may be heard of flow cytometric analysis. The bottom blue color graphs indicates the flow cytometry. Here you can see that in the control lane, there is a small peak. This small peak indicates the number of dead cells in the platform. The sub-G1 cells, we also call it as sub-G1 cells. Sub-G1 cells are nothing but dead cells. In the control, there was 4.31% of dead cells. As we proceed with the 24 hours time point and 48 hours time point, when we treated the cancer cells with higher concentration of the drug, you can see that the sub-G1 cell population increased to 38.7% and 92.16% in 48 hours treatment group. So what you understood is, at 48 hours, if our drug the cancer cells are treated with our drug, at 48 hours, it kills approximately 92% of the cells. So this is one of the amazing results that we got. And so we carried this drug further. This is another assay where again you can see the increased concentration of the dead cells upon the treatment with the drug. So here, phospho-STAT3, we wanted to know at what concentration our drug inhibits the STAT3 phosphorylation. If this is control lane, 5 micromolar, 10 micromolar, 15 micromolar, and 20 micromolar. At 5 micromolar itself, the drug started inhibiting the STAT3, and we wanted to know, compare with the available drug that is available STAT3 inhibitor in the market. That is AG490 is a commercially available drug in the market. We tested using AG490. At 100 micromolar concentration, the standard commercially available chemical is showing the inhibition, which is equivalent to a 5 micromolar of our drug. So indicating that we have a more potent inhibitor of STAT3 than the available drug in the market. Then we also wanted to know STAT3 has two phosphorylation sites. One is tyrosine 705, the other one is serine 727. We tested what is the effect of serine 27 on the CMO on this serine 727. So it had no inhibitory activity towards serine phosphorylation, indicating that our drug is very specific to the tyrosine phosphorylation. Later, we tested with the JAK1, JAK2 proteins, and also the phosphorylation of JAK1 and JAK2. We got a very wonderful results, which are on par with the results observed in the phosphostat3 expression. And we got to know that all the kinases, upstream kinases, were downregulated. Activation downregulation took place. So in this picture, you can see that seeing is believing. See, all this way I was explaining you that the STAT3 undergo phosphorylation of the STAT3 undergo downregulation. But in this picture, you can see that where exactly STAT3 is located. See, we use particular antibodies which binds exactly where the STAT3 is present. In the CMO treated group, you can see that in this picture, there is a circular pattern of fluorescence, indicating that the STAT3 is present only in the cytoplasm. Nucleus has been stained with the DAPI. You can see the nucleus here. You can see that the dark nucleus in the STAT3 lane, indicating that this nucleus does not contain the activated STAT3, indicating again STAT3 movement of the STAT3 from cytosol to nucleus has been greatly hampered upon treatment with the CMO. So if there is no nuclear STAT3, of course, the DNA binding also reduces. Then there are markers of apoptosis called as PARP and caspase 3. So there is a clear-cut activation of the caspase 3 and cleavage of the PARP protein, which indicates there is a apoptosis induction. So I think these slides can be skipped. Here, yeah, see, whatever we prove it in all this way, I was explaining to you about how the STAT3 phosphorylation is inhibited in the cells, cancer cells that are grown in the laboratory. People in the scientific community, they never agree if you just show it in the cellular conditions. You have to show it in the organism. Therefore, what we decided is we incorporated a tumor into the mice. You can see here, the mice were incorporated into the tumor. The signal, fluorescent signal, that is either blue, green, red, all these color signals indicates there is a presence of the tumor in the mouse. Those mouse which are bearing the tumor were injected with CMO 2 mg and 10 mg doses. We got a very interesting result where 4 of the mice were completely devoid of the tumor, and of course 2 did not respond to our treatment properly. So indicating that it is working well in the in vivo system also, and it may be considered for the future clinical trials. So we published this work in Journal of Biological Chemistry, which is a highly reputed journal, in fact. So this is what about the first project. In the next project, see, so whatever I explained to you about STAT3, we have been continuously working about the inhibition of the STAT3 signaling using different molecules. I explained to you till now about the synthetic molecule. So now we also worked on a compound called as bergamatine. The bergamatine is a chemical compound which is present in grapefruit. Grapefruit antandre kannadalli chakota antandre. Grapefruit contains a secondary metabolite called as bergamatine. This is the structure of the bergamatine. Since I am talking to you about bergamatine, I should tell you something that everyone of you should know. Bergamatine is an inhibitor of cytochrome P450. So it is highly advised not to eat the grapefruit along with the medications, especially such as statins. See, many of you, I'm very sure that in one of the family members, one of your family members are taking statins, because each our family has a diabetic patient, as a hypertensive patient, as a hypercholesteremic patient, lipid lowering drugs. We use lipid lowering drugs, right? So what happens is when you drink this grapefruit juice, or when you eat grapefruit along with some drug, the component that is present in the grapefruit inhibits the cytochrome P450. When this enzyme is inhibited, the drug concentration increases in the body. So basically, the people who are taking statins should not take grapefruit along with the statins, because generally cholesterol biosynthesis takes place high, and rises to the peak during the midnight time. So generally statins are taken during night. If you consume grapefruit along with the statins, it increases the bioavailability of the statin and increases the, so statins are projected to eliminate it after maybe 8 hours or so. If you eat grapefruit along with the statins remains in the blood for the longer time, and they will have a negative effect on your body. So it is highly advised not to have grapefruit along with the drugs. So we were interested to know whether the bergamatine can inhibit the STAT phosphorylation or STAT3 activation. We got interesting results. Again, STAT3 inhibited the phosphorylation, and as I explained you in the previous pictures, it showed a circular pattern indicating that STAT3 is inhibited by bergamatine. And subsequently, we also showed that baricitinib, which is a drug that is available in the market today, thalidomide, these are the drugs that are used for the treatment of multiple myeloma. These drugs also showed good inhibitory activity towards the drug proliferation of the cells. Here, the red color cells indicates the dead cells. You can see that these two are the control cells. There are less number of red dots. So upon treatment with baricitinib and thalidomide, the number of dead cells increased to 37% and 27%. When baricitinib and bergamatine were combined, it showed 70% cell death. In case of the thalidomide, it showed 50% cell death, indicating that here this bergamatine can be given along with the standard therapeutics. So like this, we have worked on several the natural compounds. That's why I am calling it as drugs derived from Mother Nature. So we have nimbuloid. Nimbuloid is derived from neem. Neem has a rich component called as nimbuloid, which has a very good anti-cancer activity. In the same way, our turmeric has a compound called as curcumin, which is widely tested against different types of cell models, which is a magical drug, in fact. So like this, we have explored many chemicals from the natural sources and implemented them as the good anti-cancer agents. And this needs to be taken further to explore more of their beneficial effects towards the anti-cancer drugs. So like this, we have been working with the drug discovery program, and recently in 2022 itself we wrote good articles related to the STAT3 signaling inhibitors from the Mother Nature. And very interestingly, I think I can speak to you about some other compounds in some other webinar where bacteria also has. So since I explained you about plant metabolites, you should not be under the impression that only plants are the source of drug. No, even bacteria can also become work as a source of the drug. So recently in 2022, I myself being the first author wrote an article on bacteria as a treasure house of secondary metabolites with anti-cancer potential. So we have discussed about the drugs that are originated from the bacteria and currently which are used for the treatment of the cancer. Like this, we have several papers published in last 2-3 years. The papers presenting here are just published between 2021. You can see that BBA reviews where we explained about what is the role of long non-coding RNAs in modulating the STAT3 signaling in hepatocellular carcinoma. We are more inclined towards the hepatocellular carcinoma. Our interested field is liver cancer, in fact. So this paper has got, as Naveen mentioned in the introduction, so this paper got the best paper award, and I have been invited to deliver a talk at the University of Paris on this paper. So this is one of the landmark discovery from our laboratory again. So where we prepared a BAD inhibitor. BAD is another protein that is overexpressed in the cancer cells, and we have a very good molecule for this. This has been patented and has been commercialized as well. So these are some of our other papers, and this is the work we patented through Government of India recently. So this is the certificate that we obtained from the United States of America for patenting the STAT3 inhibitors, and we have been taken long way through the designing and discovery of the STAT3 inhibitors. So again, it is a patent application, in fact. So our drug is commercially available, and Medchem Express is selling the molecule as a potent BAD inhibitor. This is available in the internet. So these are other important publications from our laboratory. So other than this, the last part of the webinar is acknowledgement section. I'm very thankful to Professor K.S. Rangappa. He is my mentor. In fact, PhD was, who helped me, guided me, inspired me to do all this research work. And the next one is Dr. Basappa. He is again one of my collaborators who takes care of the chemistry part of the in our papers. And the last one is Dr. Gautam Sethi, who is a supporting pillar to myself, because some sophisticated experiments cannot be carried out in our laboratory conditions, and those experiments Professor Gautam Sethi is helping us a lot. So he's because of him we are doing good science, in fact. So lastly, Indian Academy Degree College has provided me a platform to interact with so many people. 205 participants are there still. It's a good opportunity. In fact, the institute recognized me and called me to deliver a talk. I thank the Indian Academy also for providing me this platform. And I thank Naveen especially for inviting me. Because of him, I got in touch with the Indian Academy Degree College. So I thank each one of you for listening to me patiently so far. Thank you.
Thank you, sir. Thank you, sir, for enlightening us with your knowledge. It was really informative and useful topic, and also revealed many interesting facts about natural sources of anti-cancer drugs. This will definitely help students, faculties, or serious researchers in their future research path. Thank you very much, sir. Sir, now could you please take up some questions from participants?
Please, Naveen, please.
One of the participant is asking, why cancer metastaticizes?
Good. Actually, you should know one thing. Maybe for the degree audience or the postgraduate audience, these things are goes above the head. That's why I did not mention about it. Since it is a good question, I'll try to answer this. See, cancer cells are not terminally differentiated cells. I mean to say, so during the process of stem cells becoming a normal cell, during the process of differentiation, what the cells that we have it in our body are terminally differentiated cells. That means they cannot become some other cells later. But in case of the cancer cells, they are partially, they are undifferentiated cells. Therefore, they start expressing a protein, a set of proteins called as EMT proteins. See, generally, epithelial markers are expressed on the normal cells, but cancer cells start expressing mesenchymal cells, mesenchymal markers. So epithelial markers means like it could be N-cadherin or E-cadherin or something like this. So, but they start expressing a set of proteins called as occludins, viminantins. So these proteins have the potential to migrate the cancer cells, first thing. The second thing is, cancer cells express a particular protein which can degrade the extracellular matrix. As you know that in the cells are held together like this by extracellular matrix. Extracellular matrix is present surrounding this cell. So cancer cells particularly produce some enzymes called as MMPs, matrix metalloproteinases. These matrix metalloproteinases cleave the extracellular matrix. Once the extracellular matrix is cleaved, those cells have the ability to move from one place to other. So there are two phenomena taking place together. One is the EMT transition, that is epithelial to mesenchymal transition. The other one is the release of MMPs, which causes the cancer cells to move from one place to other. Naveen, can you hear me?
Yes, sir. One more question. How turmeric powder acts as inhibitor for anti-cancer drug?
Turmeric, as I said, it has the active principle curcumin. Curcumin, that is the probably the most studied molecule so far. Maybe is the curcumin, because it was tested against all the possible cellular targets. And as per my understanding, curcumin also inhibits STAT3 signaling. As I said, STAT3 signaling is again connected with the proliferation of the cancer. Therefore, this could be one of the target of the curcumin is due to STAT3 activity.
Okay, sir. Another student is asking what type of bacteria are good source of anti-cancer drug?
Anti-cancer drug. Shall I share my screen?
Sure, sir. Sure.
If you want more information, I can show you the screen itself.
Okay, sir. So student name is Bhavani Banu. So she is asking the question.
I can just show you my screen. Here in this article, we have written bacteria as a treasure house of the secondary metabolites with anti-cancer potential. Please go through this article. You will find out several, see, these are the structures of the compounds that are derived from the bacteria, purely from the bacteria. It is actinomycin D, epithelion B, xabipilone, bleomycin. Like this, there are hundreds of compounds. It is not about a particular compound. And every bacteria produces its own set of compounds. Every bacteria, I'm telling you. But we should explore whether the compound produced by the bacteria has any effect on the disease models. We should find out. See, actinomycin D, it is a drug. It is synthesized by Streptomyces parvulus. In the same way, doxorubicin, it is produced by Streptomyces pseudomonas. Like this, every drug is synthesized by a particular bacteria. So you can just go through this article, you will find out why hundreds of drugs have been discovered using bacteria as the source. Naveen, you can keep yourself unmute rather than having this gap.
Okay, sir. Okay, sir. Here some internet problem is going on. That's why I can't able to hear little bit. Fine, sir. Um, Actually, another student is asking how much grapefruit juice does it take to cause the interaction with statins?
The small amount of grapefruit is enough. You see, what my advice is not to eat grapefruit along with the drugs, because grapefruit contains lot of bergamatine, which in totally inhibits the P450. Statins has been, that dose has been standardized. It is given maybe 10 mg or 20 mg, and it should be, the dose varies from 5 mg to 20 mg. Highest dose given is 20 mg. So the statin should not reside in the bloodstream for longer time than the prescribed time. So if you eat this bergamatine along with the statins, definitely it will increase the bioavailability, which will have a negative impact on the human health.
Okay, sir. Okay. One last question, sir.
Yeah,
If
please.
if smoking and tobacco chewing are minimum risk factors for the cancer, why the government or medical council not taking initiative to stop at its production level in India or abroad?
See, these are the policy matters where, the voice can be raised against it, but it cannot be stopped. There was a issue related to the production of the aracanite. Aracanite, the aracanite related products should be banned. Immediately, the farmers started strike. Since it is the age-old practice, you cannot ask the farmer to cut all his aracanite trees and leave the land, because it is the source of the income to them. And the people are being educated through, if you see this cigarette pack, it's clearly written it causes the health hazard. It is like you are allowing the farmer to grow, you are benefiting the farmer, at the same time you are educating the smoker. So it is like, if you educate the smoker, it is the choice of the smoker, but for farmer it is life. For the smoker or those who eats it, it is the choice. So whether he wants the life or not, though you know that it's bad, but still you want to go for it, go for it. The government is getting enough revenue out of it. Why the government wants to lose itself?
Okay, sir. Thank you very much. So I really thank you again for enlightening us with your knowledge, sir. So now we have one small session at this end. So this event from our students. So that's why I'm handovering this session to the Suma madam. Madam, it's
Please, Nan, please.
Thank you, Dr. Naveen, for conducting the session. Thank you, Dr. Mohan, for such a wonderful talk today.
Thank you very
We
much.
would now like to take you. Yes, sir. Thank you so much, sir. We would now like to take you all through a small virtual cancer awareness tour of posters. This has been created and contributed by our 5th semester BSc students. Our presentation is starting now. So are you able to see the presentation, sir?
Yes, madam. Yes, ma'am.
Okay, thank you.
Madam, I assume that the slides are not moving.
Yes, I think that is a little problem. Just a minute, please.
I appreciate the efforts of the students as well as the teachers in preparation of the presentation, as well as the institution is doing good job by enlightening students with this kind of activities. They should know, apart from the curriculum, it is very important to know what's going on, how exactly the research is being implemented. I think you guys are doing good.
Thank you, sir, for your kind words. Now, we are now at the end of today's event, which I hope has shined illumination on and given clarity about a major global health issue. I would now like to propose the vote of thanks and express my gratitude to all the people who have assembled today, as well as to those who have worked behind the scenes to make this event a great success. First and foremost, I extend my heartfelt gratitude to our esteemed chairman, Dr. T. Somashekar, and our respected principal, Dr. Srinidhi K. Parthasarathi, for their constant support and encouragement in organizing such an event. I also extend my heartfelt gratitude to our eminent guest, Dr. C.D. Mohan, University of Mysore, for sparing his valuable time and providing a very lucid picture of a global problem and the attempts to treat it. I wish to thank our head of the department, Dr. H. Anandavardhan, for his immense encouragement, guidance, and involvement in the conduct of this webinar. I would also like to thank all my department colleagues for their unstinted hard work and cooperation extended for the smooth conduct of this webinar. I also actually thank the dear students of 5th semester BSc who have used their precious time during this lockdown period and used their creativity in coming up with such a great way of providing awareness. We greatly appreciate your interest and involvement. My gratitude also extends to this large gathering of students and faculty. I hope sincerely that they have greatly benefited from such an illuminating and wonderful view of a global disease and its possible local treatments. Last but not the least, I extend my thanks to the IT team for their support in the organization of this online program, and also to the design team for their assistance and cooperation. Thank you, one and all present here, for attending this webinar and striving to make this a grand success. Dear participants, feedback link is posted in the chat box. I request all of you to kindly fill it to get your e-certificate. Thank you, one and all, and have a good day.
Thank you very much, all.